protein simulation predictions and drug responses and even clinical phenotypes data management. So we all come together and realize that our area of focus should be on the effects of predictive effects of all of these data set from all of these areas towards predictive genomics and also personalized medicine. So so we have come up with the main vision and challenges that we have for personalized predictive genomics. So from what we discussed, from what we wonder, right, is it from medical records like traditional diagnostic clinical data, genetic data sets, and multiple omics data set like RNA or protein profiles? How can we collect, compare, standardize, and integrate these data sets into a collective interchangeable AI model to predict clinical outcomes, drug responses at a personal level? So it's the definition of personalized medicines. So in order to, because we realize the main challenges in this field, right, what we have is a limited amount of information for prescribing drugs to patients. So also every patient and every person in the population, right, they have very different specific genotypes and also have different genotypic environments. And the mutants, that is, even if we identify the mutations and variants in the patients, right, they can be expressed very differently in every different patients and different person. Then, so what we're trying to do is to try to integrate multi-omics data, clinical data, phenotype data to solve this, and then to try to come up with a predictive model to better improve and optimize personalized medicines at a single patient level. So the approaches that our group propose is, first, we need to ensure that we have a good quality data set from all levels, that for all the integrated levels that we have discussed, from genomics to proteins, also to phenotypes and outcome data. So by good quality, by good data set, we means that the data set should be very well defined, very well QC'd, and very well labeled. From there, we can train AI models on systemic data integrations. So with refinement from very good complex existing data set, so-called like supervised AI model. And also after that, we can follow by longitudinal studies for further integration into AI for further like validations for our AI models. So, so the requirement for our data set is we need very good cohort for training and can be applied to Singapore. And also we recognize one issue is that not really issues, but one questions. So can we actually extract features from small cohorts, let's say in Singapore, to build predictive model as a proof of concept study, and then broaden the applications to like general AI applications? Yeah.
Okay.
Yes.
That's all?
Yes, that's all.
Thank you.
Thank you.
Sounds interesting. Any questions? And if there are any questions, we assume we're great to help.
Thanks for the beautiful summary of our chaotic ideas. I just wanted to add one comment. A lot of this was in response to Joanne's rap on the knuckles saying, do something new. Don't just ask for a lot of money for a huge cohort. So our hypothesis and our proposal is that in addition to the unsupervised or self-supervised feature extraction that Melee was talking about from AI, can we do some supervised feature extraction and then really reduce, improve the predictions at low cost, right? So obviously your input is your massive data set. What is your most massive data set? EHR is a massive data set. Increasingly, genetics is a massive data set, right? So that's the input to your model. The output is, disease risk, drug response, something like that, some clinical outcome. How do you go from that input to the output? You can use an unsupervised feature extractor or self-supervised, say, like embeddings that AlphaFold uses or stuff like that. Or you can say that, okay, out of my massive cohort, I will spend a little bit of money on a tiny sub cohort. Let's say out of 100,000 individuals in SG 100K, we'll spend money on generating multiomics data from 2,000 individuals. Let's say just 2,000 out of 100,000, 2%. But that multiomics data will be, will be part of the chain of causality from genetics to outcome. And so it can be used to supervise the feature extraction from genetics. So basically, instead of you going as one flying leap of predicting outcome from genetics, we predict RNA from genetics, and then the features that we extract in that process can be used to predict outcome. So a two-step model. And so perhaps this way we can really improve the quality of our AI with very small additional cohorts, rather than improving quality of AI by saying instead of SG 100K, we'll make it SG million K, which is 10 times more expensive. So that was the general philosophy here.
I'm interested in the responses of the computational scientists in the room, those of you who are still here, in terms of that part here of like what models may be useful. I'm not sure whether there's anyone else here want to add to that.
Okay, I think I can add one more point is that we can heavily apply a language model. For example, even for the electronic health record, we can use or medicinal language model to embedding, and the bottom, the genomics, we can use the DNA language model, and such model, right, is easy to integrate and cross attention or something like. With this, I think it's a doable that we can combine all the data, either using the supervised learning, which we discussed, we have maybe small cohort, but very clear label data, which we can assign together, and not very extensive, but very focused. Then I think we can use this AI tools to come up quite a reasonable, supervised learning model based on multiple language model on all together. I think this all may be the.
I think it's interesting, and there are some new joint embedding models that can be used here. And I think it is also a good introduction for Sebastian's workshop of biomedical data. So how we can extract data from medical records. So can we use the ChatGPT and how to provide this? So Sebastian, do you have any suggestions?
Just to share, we are doing some of this at the moment with SingHealth data. We went in to look at phenotype data. For example, I'll just give you a very quick example. So for example, ER/PR positive, HER2 positivity. So for cancer people, we need to look at some of this immunohistochemical test results, right? So what happens is when you go in and you will see there'll be lots of gaps, the clinicians are reporting them all over the place and very differently. We've been using language models to just extract the data, ask the language models, what is it most likely to be? And then we keep training that. And then it's actually really, we've been able to plug all these holes. And so what ended up being a very holy data became quite filled up. And I do think there's a very good way, one example that's already currently done. Yeah, but I'm sure Sebastian has other ways.
No, I think you are right, and there is a lot of opportunities using these new models. There is one complication that training on new models may be prohibitive to do on the Trust platform because of the compute required and how much computer is actually connected to it. However, many of these things, also if you have your own code and if you have your own data, this could be explored outside, and then when it's stable and established, brought inside Trust as an algorithm. And there are many opportunities to take, let's say, pre-trained models and fine-tune them to specific different tasks. And I think also what the community should consider is not everyone making their own model, but rather have a good awareness of is there one bigger model available that more people can use inside Singapore?
Yeah.
Yeah, any other comments, critiques actually of first group's kind of suggestions? Again, we're also looking out for Zoom questions. So if there are, please type. Thank you for writing the paper for us. Okay. Is it though? Okay, so hi, in our breakout session, we discussed creating AI research inspired by genomics and achieving sovereign genomic data infrastructure. And in the scope of this challenge, we identified several sub-challenges, and we envisioned them as a way of achieving sovereign data, genomic data infrastructure with the assistance of some AI approaches. Namely, we wanted to discuss the ideas of developing a method for representing the genomic data, including correlations to the phenotype while preserving the anonymity of the genome. We also discussed how could we generate useful synthetic data that could be used in the training of the AI that would compress and represent genomic data. All of this, of course, is related to privacy and security of the genomic data for humans, but we also discussed the problems of the current data that we have, which is probably not well suited for developing AI and storing it for future analysis, as well as differences in privacy and security concerns that we have for human data versus other organisms, for example, plants or pathogens. And maybe Laura can continue.
Thank you. So yeah, one of the main impacts of this work is in facilitating data sharing and enabling research collaborations by allowing the processing of raw genomic data in combination also with the phenotype metadata, while ensuring the privacy of the individuals. It's also worth mentioning that in some cases, this privacy of the individuals is not so important. For example, if you're working with plants or pathogens, but then in some cases you have different considerations that you need to take into account, such as, sorry, not too fast. Such for pathogens, we mentioned like maybe back engineering their genomes and how much of this can be made publicly available. Or some private companies developing commercial products based on public research data without acknowledging or compensating the public efforts. In terms of models, the genomic-based models face unique challenges that would need to be addressed in the design of these AI solutions. And we see the diffusion models and transformer-based networks as good baseline architectures on top of which we could develop some models or AI solutions specific for genomics. While these diffusion models and transformers are usually developed for text or images, we anticipate that the genomes bring another level of complexity. For example, size of the data, sentences on which transformer models work are short, even whole paragraphs that you can feed to GPT models are fairly short, at least when you compare them with the size of the whole chromosomes or genomes. Also, another key point is like how to ensure perseverance of relevant information and enabling meaningful processing. For example, genome is a lot more context dependent compared to language. We have the same DNA maybe in the brain and the liver, but the transcriptome is completely different. So there's several layers of complexity, whether we would be working on the genomic data of RNA sequencing or transcriptome data. And that's all aspects that are not really addressed in the current off-the-shelf models, but would need to be incorporated in the models specifically for genomics. Also, a lot of data currently available, we believe, is in like tabular, even though that format is perhaps suboptimal. So reformatting this data as graphs could be beneficial for easier processing and easier inference. And also one of the points that we discussed is the use of and the generation of synthetic data for training. Real data can be scarce. Sometimes it's difficult to obtain it, or you face issues like privacy. So is it possible to generate synthetic data reliably so that the distribution of this synthetic data follows the real data? Which scale? How much of this data can you generate? And I guess like then you could maybe use real data only or either on inference or for fine tuning on it. And standardization of metadata is also an important aspect. For example, can AI help here? Because a lot of metadata can come in various forms where it's not standardized, and then you would have to implement a specific module that would parse this data and extract information that's useful for you. But perhaps AI could be beneficial so that it can just summarize or extract useful knowledge from this unstructured the type of text or tables or whatever we are dealing with. That's all from us. Thank you all for listening.
Great, thank you. So we need to prepare 3 to 5 pages. You already prepared 2. That's good. So there are many interesting topics, and I think that one of the most important is how to enable training while keeping the privacy. So probably some kind of federated learning and this kind of approach will dominate in the field. And yeah, I'm really eager to hear AI experts, so how they will sort out this problem, because I believe this is one of the problems. And also, so many people talking about DNA and language models, and so just question for you. Do you think that it's worth involving information about methylations so that we have actually the 5 letters instead of 4 in language models? So if anyone has any comment there, please go ahead.
I want them. For
Sure.
cancer, I want them. For aging, I want them. So
Well, Nicola.
Yes, obviously, but probably no, in the sense that like methylation is dynamic, while the genome itself is not. I mean, unless you have a cancer, but and so that's probably a bit tricky. It's probably both, I would say. I mean, like one approach that consider, yeah, and then maybe more than just methylation, but sort of like a like histone mark, like a much richer one. But then again, it's dynamic, and so it adds complexity.
Yep, I agree, and it is interesting to take care about heterogeneity in cells, so that we have several kind of cells will probably have different methylation patterns.
I'll just share that in the clinic, the clinicians want this data because it's most proximal to when they're seeing someone. So genomics, is germline along the way. If there is, for example, the epi-H that I work with Ngeow on is kind of one of the epigenetic marks, right? So clinicians want that. They want to know whether at that time point with everything else there, can you have a methylation signature that is most proximal to their condition? So there's a great, but then of course the challenge is then, as you say, because it's so dynamic, you'll be taking multiple time points. And then how do we, sort of consolidate that data? So from a clinical perspective, we want that data, but the question is how.
But one of the things can be interesting to combine some RNA expression levels with methylation patterns and then try to learn as a model how to actually, can we do some moving from one domain to another domain?
Yeah, and then also, I mean, dynamic, not just over time, but over cell state, and then like more, I mean, in the context of the clinic, we use blood as a proxy, works often, not necessarily in many other cases, I suspect, yeah, where just not the same methylation in the liver and the brain.
Yeah,
and then a brain biopsy is probably not something applicable to the clinic.
Sure, I agree. But I think that just epigenomics is a very, very important topic, so that when we are thinking, we don't need to forget. Okay, Any other questions? And if anyone, no?
All right, team 3.
Okay, thank you.
Hi, everybody. I'm Alvin. I'm from LKC Medicine. I'm a presidential postdoctoral fellow working on genomics, long-read sequencing. Yes, complex. So essentially, our team was tasked to sort of figure out, what are the challenges to look at complex diseases through AI models. But at the same time, the problem itself is actually quite complex. That's why I would actually start with the end in mind. So essentially, what we want as a vision is we want to have a very integrative data set to identify different latent relationships. I'll explain that a little bit more after that. Essentially between genomics and other exposures, because we have clinical data, we have diet and things like that. But we think that we can push it even further. So you can actually collect more information to look at relationships in the long run. And I think one of the main things that we wanted immediately was to look at relationships in a very heterogeneous data set, for example, in Singapore. So essentially, are there any ethnic differences between the groups? Can we actually detect them at this point? And that's why the short version of the story is that we don't have the data yet as well. That's why one of the things we really want is to be able to determine exposures throughout time. Maybe having methylation is an example of that, because at the end of the day, everybody's genome is static. It's sort of set when you're born, but you don't actually know what happens and why some people have higher risk of disease. And that's something that currently with data we have, we don't have that yet. And I think in our perspective, really, AI can help us with that because we cannot know the relationships that we don't know yet. We base a lot of our understanding, a lot of prior knowledge on papers. So if there's something new that comes up, we can't figure that out. So essentially to draw relationships between very diverse topics like air quality and the risk of lung cancer or lung disease in your life, lifestyle diet changes and cancer risk, colorectal cancer, for example, in Singapore, and other like sort of sporadic or instances that we can't account for where you have some sort of temperature fluctuations and changes in sort of even zoonosis and infections, for example. That's why at the end of the day, I think there's a lot of room for AI for predictive modeling, but we don't have the data for that yet. That's why at the end of the day, I think a lot of us really focus on how to get a good omics data set. It might not be just DNA sequencing, RNA sequencing, methylation, but sometimes having more information in clinical information, lifestyle, other exposures is really important. And one of the biggest issues that we have is that actually there is very poor overlap. If we collect data from a cohort, you want to have all the fields like filled up if you're a data scientist, for example, but in real life, you don't actually get that. You can have cohorts that are very deeply phenotyped, but some with none of that information at all. That's why at the end of the day, I think it's still trying to figure out how do we get data together that is cohesive, and for us to ask questions that you wouldn't think that you'll be able to ask, for example. Another large conversation that we had was around like sort of the technical effects, because everybody who has tried to sort of glue together DNA sequencing and RNA sequencing will know that it's not simple. It's really difficult. There are a lot of technical difficulties, and even how do you sort of combine that? And I think one of the conclusions that we arrived at was that essentially there are tools that can do that already. So you can have embeddings that handle missingness of data. I think one of our Siri mentioned that. And the idea is that a lot of the technical difficulties or sort of batch effects, for example, can be handled by AI that we have not explored that, but essentially that might be something really useful in our daily research life. At the end of the day, yeah, if we are at a point where we're still trying to integrate WGS and WTS, yeah, so please use AI and see whether we can actually help to figure that out, because at the end of the day, that's just the first step. That's why I think the other big theme that came up in our discussion is that essentially you can't know something you don't know. So you can't ask questions that you haven't thought about yet. But at the same time, there'll be some sort of relationship, some interaction between different things in the data set that we have in terms of genetics and other exposures. That's why at the end of the day, we need new tools that can pick out these different relationships. And for example, even if you have longitudinal data, you can use things like knowledge graphs for that. That's why at the end of the day, I think if we can handle the technical parts to handle missingness and essentially to go to the next step to find relationships between very complex data sets. One of the things that was brought up is that essentially if you do sequencing on different machines, that can even have some sort of technical artifact effect. But at the same time, that's something we need to handle, because if not, you can't get useful biological insights from that. And that's why I think one of the discussions about aligning data types and technical difficulties, essentially, that's something that is worth sort of exploring on its own as well, because if we can't get it to work for our own research projects, you don't want to implement it in the world, for example. And that's why really to get past all the technical difficulties, then we can actually work with the actual biology or the disease or do the predictive modeling we want to. Okay, so yeah, one of the main questions was, can you resolve the existing solutions? The answer is yes and no, because essentially, yeah, like I mentioned, you have models that can have different data types, you can have the longitudinal data, missing data, and then there are already examples of even just looking by a pathology slide. There are sort of models that can determine what mutation your patient has. And that's something that is entirely different and it has no knowledge of genomic data, for example. But at the end of the day, we realize that data sharing is one of the biggest issues, because if everybody is working in a silo, if you're collecting the data and not sharing it, there's no way for us to draw the sort of interactions or correlations between the different data types. And that's why we need really to come together, put our data together and really have it harmonized in a very, very nice way. And one of the other main things is that we really need better coverage of individuals. So we have cohorts with very deep phenotyping, but for other cohorts, you might not have any of those phenotypes collected. And so at the end of the day, we might need a way to have a very good harmonized data set of essentially a cohort that is captured for most of the clinical information or different sequencing methods, and then use that to figure out whether we can even do some sort of benchmarking or some QC type thing. Lastly, yeah, we had quite a bit of a discussion on digital twin, and we are still thinking of whether this works in this case, because at the end of the day, if we have mostly missing data, we don't know the assumptions that we're working on, the digital twin might not be the best case for this case. And that's why right now, I think we sort of think about gathering even more information, more databases and more data types to start with. And yeah, the last one is, are we the right people? Of course, without a doubt, everybody here, because even within the table itself, everybody had very different experiences. We have epidemiologists, computer scientists, computational biologists, and that's the first step. You need to get everybody to sit here at a table and really figure out what are the issues. And so at the end of the day, yeah, please talk to each other, share your data, and I think we can think about more interesting solutions from there. Oh, thank you.
Okay, thank you. Many topics. So I have a question as a computer scientist for you. So you mentioned at the beginning environmental influence.
Yes.
So how you can actually measure this influence? So for example, maybe we can have some somatic mutations, epigenetics, but is it possible to see, for example, some epitranscriptomics so that we have some RNA modification, because there are many, I don't know, 150 RNA modifications. So I'm just trying to ask question, what kind of data you can produce? And the computer scientists really need biologists that we understand what is the actual problem. And when we need to construct the digital twin, okay, that sounds perfect. We have some digital twin, but what does mean the digital twin? Do we have now some analog twins? We have.
So, okay, so I think the first question is RNA modifications. I think we don't go there yet. I think we'll start with methylation, because with some diseases, for example, if you have poor air quality or you have other lifestyle changes, epigenetics in the blood might be the first thing to look at, because at least you can get it. You don't need to go to get a biopsy from every patient, or if individual, actually, healthy individuals. So I think that's the start. But also, I think the second question really is, yeah, that's why in our case, we don't know about the digital twin. We need to identify features first and to really define a question, because right now we are exploring sort of the unknown hypothesis. We are generating data, generating hypotheses from there. But if you know that, okay, it's air quality, if we really want to just look at methylation, whether any changes in COPD, for example, there might be a way where you actually want to use the digital twin.
So just maybe to broaden the discussion a little bit. So a lot of these things you say is unknown. We don't have the longitudinal data, this and that. I just want to broaden the conversation a bit more to say, can we use plants as a surrogate for now for methods development and then apply? I'm just putting it out there. Is that a feasible approach? Look, I make the see the hand went up.
Yeah, I was about to comment about this. So I think There's recent development on plants that so these high throughput phenotyping platforms and all that. So you can actually try to connect the genotype to the phenotype in kind of a methodological way in plants. So I think then if you can do that in a rudimentary way, then you can try to add more layers. So RNA-seq methylation and all that, and then try to get improved predictions and so on. So I think that could give a template of how to construct this model. So what kind of architecture you should use, and then you can apply that to humans where there's kind of sparsity of phenotype data. Then
I did promise to take care of the plant, guys. Okay, come. Sham had a question.
I think environmental exposures are very relevant and it's part of getting better data for our AI models. Traditionally, it's just been hard to systematically collect a decent amount of environmental exposure data. And that's certainly something we need to grapple with. But I wanted to also come to Mele's point about how do you know what's mediating those environmental exposures? How do you know that the body is responding to those environmental exposures? You mentioned DNA methylation because the traditional approach is that the environmental exposures change the epigenome, they don't change the genome. And for the epigenome, there's a kind of a mental reflex that epigenome equals DNA methylation. But actually, environmental exposures don't just change DNA methylation. They change gene expression, they change proteins, they change, even tissue architecture and so on. So I feel like to have a competitive advantage, Singapore should probably look at in a more holistic way at the impact of environmental exposures rather than DNA methylation alone, because everybody's looking at DNA methylation. Yeah, that's just my comment.
Yeah, no, I totally agree, because at the end of the day, yeah, I'm not an expert in the field, but also I can imagine if we use models that can pick out patterns in the protein sequences or in RNA expression changes across time, that'll be really interesting.
Okay, I think we need to move on.
Yep.
So our topic was advancing generative and synthetic biology, even though none of us from the group are actually in this field. So this is more maybe not that specific as previous groups. So I think when we mentioned this topic, all of you are aware that what we want to do here is to engineer sequences with some desired properties, meaning how can we create certain promoters or enhancers or certain ribozymes, and to create sequences which will create novel proteins and even creating and even more some organisms, and getting this to even a higher level. It's also interesting to know like how mutation of every nucleotide could impact organisms and what can happen if there are some changes in the all of the nucleotides. There are many impacts where this can be useful, as you're all aware, but some things that we mentioned is like impact in the agriculture. How can we create maybe more nutritional food, or like if we can create new vaccines which could be helpful. Also regulatory functions of RNAs, proteins. Can we create some RNAs, proteins which could do some regulations, especially in certain types of cells, not in all cells, but in specific cell types. Okay, but with generative AI, we come to some huge bottlenecks. The main one is like, how can we tell that something that generative AI created is meaningful? We can use AI to create a deep model to create a new RNA or protein sequence. But how do we know that this sequence is how can we be sure that this sequence can be stable? Like, can it even exist? And especially, can this sequence do the function that we wanted it to do? The best way to check some such things is, of course, experimental. But with experimental checking, this is a huge bottleneck because it is often very expensive, time-consuming, and sometimes it cannot even be done. Like as mentioned in previous talks, like if we want to have some regulation in neurons, like how are you going to check this? How are you going to see if this is really useful? So we believe that even if you are able to do the experimental confirmation that something is working, that it's very important to have this iterative process between AI models and experimental confirmation. So when you confirm something that it's working or not working, use this to further train your models and to obtain some new knowledge with the AI. Yeah, as for the models that are useful in this area, definitely diffusion models, which come from noise and create some meaningful sequences or whatever. GNNs, which are, again, used a lot, especially for the structural part of these sequences, like how do RNAs and proteins look like. They can, of course, be represented as graphs, but can we connect this even further? Like, can we go from sequence, our DNA sequence to RNAs and proteins, and is there a connection, and can this be represented as a graph as well? Foundation models, of course, and there can be specific for foundation models for DNAs, foundation model for RNAs, but also can you combine this as a multimodal, having a multimodal model that can use all the omics. Also, as Mila has mentioned, I think we agree that it's really important to design new models specific for genomics. Like, can we, because like all of the previous models, GNNs, CNNs, they were all designed in a way that they integrated certain biases into these models. So can we come up with biological biases which could be used then to further design such models? Some ideas that we discussed is that it may really be useful to combine omics data. So for example, like if you have DNA sequence, then can you use it as a simple DNA sequence of nucleotides, but then can you convert it to protein? Try to use this as well. Of course, here you have a problem of what is your starting point. But even if the generated data is not correct, maybe if you have enough such data, your model could distinguish between useful or not useful stuff. Yeah, and of course, we are coming back as all of the groups to data-related problems. We always need more data. We need the data to be more quality, so that it's more that has higher quality. We need to understand what noise is coming from wet lab experiments, what is some computational noise that is obtained from the methods that we have in base callings and stuff like that. We also want to emphasize again the importance of sharing the data, exchanging the data between labs, and also mention that, as it was mentioned in the previous talk, that metadata is also very important. That is, and the reason for that could also be that not just that it's poorly collected, but you don't know what you need to write down when you start certain experiments. And we think that AI, not in terms of deep learning and the models, but like simple ML and AI could help with this as well, in terms of quality assurance and collecting this metadata. For example, reminding people in wet lab to write down things or automatically save information about chemicals used and stuff like that. And again, coming to the point why we write people to do this, we believe that Singapore has great governance of data, which could help with collecting and sharing this data. We also think that it's a very diverse community as in terms of human genomics with a multi-ethnic culture, but also if we are looking at biodiversity, which is very high in the tropical climate. We think that there are already many sequences available in projects such as National Precision Medicine, and that we could leverage these data to create new foundation models and other AI.
Okay, thank you, Joanna. Questions?
Question is, how do we scale? More data. I think that's always the answer, that we need to produce more data and then see what we can do with this data.
And there's maybe another group of workshops in the future. It's going to be robotics. So how to use robotics actually to produce more data. Probably it's going to be one of the, in future, we'll see soon. And also, I believe, and I briefly discussed that more with the group 5, is that, you know, for language models, for image, all of us are actually good in some way, that we can recognize what is the good text, what is the good image. Okay, some of people can see in the gallery something that I cannot see, but most of us can have this feeling. But it is specific for this kind of problems that at the beginning and at the end should be biologists. So I think that that is one of important things that people should understand, that they are not, that not can everyone check what is correct results at the end or not. So we really need to have wet lab people who can prove that something actually works. Okay, if there is no, is there any questions from the Zoom? How many people are still there? 3. Okay, 37. Okay, almost the same number as here. So please, all of you, turn off your mobile phones and Zoom so that we can see how many people actually are on Zoom.
Probably very sustainable. Getting that model data. So I'm not sure whether there are other ways. So I'm always quite inspired by, you know, every time you enter something, you know, the capture, you know, all this kind of going into, you know, image analysis. I'm just saying, like, how do we, is that a way to other things that help us scale rather than me when, you know, select longitudinal data, etc. We've been doing that for 30 years. At the base of whatever that is. It'll take us a long time to get there. Using cardinals, other ways.
Yeah, so I think one approach is we just do more of what we're doing, or there may be in some applications it's not possible, right? And we have to be more targeted and then say, actually, these experiments are the most valuable ones from the perspective of training a model. So I think using the way we think about modeling the data to guide how we do the experiments can be very valuable as well. Because some of these bottlenecks cannot be overcome, right, in terms of the number of experiments that we can do. So then thinking about how we do the most effective experiments, generate the most valuable data can be useful.
Maybe also a way to scale. I think since it's all about like acquiring more data and higher quality data, is to actually have people engaged and then willing to like turn in their data, like feeling good about turning their data is something they benefit and something they do not need to fear. And so that can be, so as we sequence genome, right now, this is paid by the taxpayer, but eventually that could be something like people would be willing to like shelve personally the $1,000 it costs to have their genome sequenced if they can have some benefit in turn. Maybe people would see as beneficial to them to like turn in their Fitbit data, their Instagram of like the food they eat every day picture. So that like the flow of information and of good quality information, can be acquired sort of at cost, at scale. But for that, there is really an important component of like people need to probably like see a benefit to it, and also not fear that this is gonna like there's eventual nefarious use of it, because it's a one-way thing. If you turn in your data, you cannot claw it back. And so yeah.
Thanks, Nick. I think it's a very, very good point. So there are lots of data, but do we share this data? And can we share data? So it is again of the privacy problem, but something that really helped computer science and AI was that people shared their code. So it is easy to reproduce. They shared papers. So all of us put first on archive or bioarchive so people can read it fast. They don't need to wait 2 years that someone read. So how to speed it up? So if we want in biology to increase the speed, we need to know how to share data and how we can reproduce results. So yeah, I think this is important questions. Any other questions here? Okay, thank you, Joanne. Oh, there is one question for chat. Oh, the question is from Chandra. We need someone who is a specialist in ethics in AI who can advise and audit data that is being used to build models. Do we have one? But there's two things. So it's good to regulate things, but if you try to regulate everything, so that will stop research. So we should be very, very careful to take care of privacy, but not to go too far so that that can stop all kind of research. So this is just my opinion. And yeah, there are always discussion. So what can be the potential problems of development of sites? But this question is much older than us. But the point is good.
Okay, so I think our discussion was mainly filled with frustration in the end about the lack of a genomics-aware workforce. And I just want to share an example. So I've worked in a wet lab for many, many years as a molecular biologist. And I swear there wasn't a single day where there wasn't a job advert out there. We're looking for computer scientists. And I swear, and then when we had people coming for an interview to join our lab as computer scientists, we would literally roll out like a red carpet so we can like lure them into the lab. I'm just really hoping they would join. And it really depended on the institute. In one institute, then we had a great call facility, so we could hire bioinformaticians to kind of work for us. Obviously, we would pay them. And then often the language didn't work. We just didn't speak the same language. And then in another institute, we didn't have this core facility. So we kind of just had to hang around and wait for someone to come around who could do this work for us or just collaborate with other labs. And that obviously always delayed projects, delayed data analysis. And I think the two main questions are, us as molecular biologists, we should be able to do some of the simple coding ourselves so we can just get on with our work instead of having to wait around. And then those computer scientists, where are they going? Why is no one applying to these labs for these positions? Are we losing them to like other tracks like finance or hedge fund or some of these examples? So this is where we thought kind of the main issue lies. So we saw it mainly on a human level. So we believe we kind of have to start like at the bottom, at the education level, to kind of expose young people from a very young age to both directions, that they can be computer scientists, but also have a genomics background and vice versa. And also for these computer scientists with a genomics background to create a career path for them, so they kind of know where this career leads them. So we don't lose them to finance or to anywhere else where they just get way more money than they would in, as we know, in academia where we are. So we want to create a multidisciplinary workforce to create this workforce, to create AI solutions with the right skill set. And these are just some things we mainly talked about. We want to prevent that siloing early on to build that team, and update curriculums, and starting very early on in schools, at undergrad level, to update curriculums to kind of incorporate both. We need to improve that communication. As I just said, I didn't really know how to talk to these computer scientists, so we need to improve that communication so that we speak the same language. Knowledge. Biologists, they often don't even know the strength of AI and what we can actually do with it, how we can use it, that helps our projects. And the other way around, as computer scientists, they often don't really seek those biology experiences. So again, how can we lure them in? We need more exposure, more internships, more collaboration with industry, and mixed program that incorporates both. And this could also involve teach coding from primary school from a very young age, so that when they come into a job to a postgrad level, they already know how to code. We don't really need to seek out expertise from somewhere else. Get girls at a young age into computer science, that's a question mark. I don't know why, because that's a fact. Yes, we do. Also change the mindset. We also discussed, and someone mentioned that bioinformaticians, they're often seen as a service. And I mean, I kind of just said I've used them as a core before, but we need to change the mindset that we're not just giving them money as a service, they're valuable contributions with their own projects as well. I've mentioned that we need to make a career path for these computer scientists in genomics, to give them a career perspective with a clear plan, so we don't lose them out on any more high salary positions. We also need a balance. We kind of talked about, we have a lot of medics that go into AI because they're kind of often more mature and more entrepreneurial already, but they often don't really know what exactly it means. And then computer scientists, they don't really seek those opportunities themselves. We talked about access to data sets. It would be maybe a good opportunity to make that easier. I think we talked about that earlier, so that students or young people early on, they can kind of play around with data sets and kind of get a better feel for it. And not just for young people, for undergrads. As we said, it's so difficult to get access to data. So I think that, yeah, we need to make that easier. So potential impact, we need to have the right people to create those solutions, because you might think, now I've talked a lot about humans, we kind of want to hear about AI, but we believe we need the people first, the workforce who can come up with AI to replace those humans, and who can then streamline the process to make data analysis more efficient. Like I said earlier in my example, it just really delayed projects. So to just streamline the process a little bit more. What types of AI might be able to solve this challenge? Well, we struggled a little bit there, as you can imagine, because we mainly have this on the human level. Create a science version of large language models. So we kind of need scientists, though, to kind of feed those language models with data. And I'm not sure this fits rightly there, but to kind of lure in those computer scientists, they love competitions, apparently. There are so many competitions out there with cash prizes for computer scientists, like AI for biology, who can create the best model, and then they get a prize for that. Apparently that might be a good solution as well. Can this challenge be solved by anything that's already around? Well, you can see by our answers of, we got more and more annoyed by this problem. We said, no, we believe it's more of a structural issue, not so much of an optimization problem per se. It's more on the education level. And then, are we the right people to tackle this? Well, yes and no. I think we need to start with on the education level. So we believe we need to talk and work with MOH education together to solve this. And the NRF here can help create this career pipeline as well that I talked about with MOH education, and NRF as well could help with the funding here.
So I'd be curious to hear whether you think AI is already here and we can really jump in there, but we've kind of seen this from a human perspective, to be honest.
Thank you. Before we start with question, I will do one experiment. So Tim is a postdoc in my lab. So he's computer scientist by background. He did a PhD in computer science, and I actually chased him. He didn't apply for my group. And so Tim, can you tell us, so what do you think, what is the problems for people from AI in biology? And does working in this field ruins your chances to find a job in some other field or something like that? But I think there are some kind of concerns people have. So yeah, be honest. Yeah, and sorry for.
Yeah, thanks, Mila. Yeah, I don't know. Actually, doesn't ruin my chances in other fields. I hope not. Yeah, I mean, when you came to offer me this position, previously I was thinking of going to some probably computer vision groups and something like that. But I think when you presented me this opportunity to work with you and in genomics and everything, I think it's really interesting. It's maybe some to implement AI in some other perspective. And yeah, I don't know what to say. And yeah, of course, it's much harder than because we need to know biology, and then there's a couple of months, probably year, to learn it, and then to start to produce something. Yeah, definitely.
As you mentioned on your presentation, we have also talked about like the communication between a computer scientist and biologists. And then in meetings, it's often very hard to, you think you understand each other, but in the end, sometimes after even months, you find out that you haven't understood each other, and that, for example, computer scientists believe that the data that biologists provided is certain way, maybe that it's exactly 0 or 1, or that after months you figure out, oh no, that these were probabilities and the data was not so certain. And the other way around as well. Like sometimes biologists trust too much the outputs of the models that computer scientists produced, and then this leads to other problems.
Yes, exactly that.
You are physicist by background.
I was going to draw on that. So there's an analogy. So it's actually a very close analogy. How do you get computer scientists to care enough about biology, to learn biology, and to understand how they can be useful to biology, rather than sitting purely on the math algorithm side? And I had a similar kind of epiphany as a PhD student in physics when I was modeling particle accelerators. And I thought of myself as a math guy, not as an applied physics guy, really. And I preferred the models to the reality. But I realized that the people sitting in the particle accelerator control room didn't really care what I said, because I was not saying anything that was relevant to them. I didn't know what knobs they had in the control room to control the accelerator. I didn't know what oscilloscopes they had to see how the accelerator was responding. So all the theory I was producing was irrelevant to them. And actually, I looked around the building where I was, and I realized actually half the people in that building were like me. They spent their entire careers, they were 60 years old, and they had never said or done anything that the people in the control room cared about. But they published in good journals, and they had thriving, successful careers. But then a small fraction of the people in that building actually did modeling that acknowledged that you have only so many knobs in the control room, and you have only so many readouts, and these are the things you care about, these are the things you don't care about. And I wanted to, for whatever reason, my personality, I wanted them to listen to me. And so it was a long journey to learn the engineering details of a particle accelerator to make my modeling useful. And I think that served me well as a computational biologist when I changed fields, that I wanted to learn the biological details to make my modeling useful in biology. And so I feel that if we can induce computer scientists to make that jump from, I will spend my whole career solving Kaggle competitions, like some of the competitions you referred to in your slides, to I will try to understand what actually needs to be done. I will try to understand what knobs biologists have and clinicians have to modify the patient's journey and what they can learn, and then address that problem rather than a Kaggle challenge. Then we've won. How do we get them to care? And it's really a change in their career trajectory. And I think if we can somehow do that as a community, that'll be extremely valuable.
Add one quick thing. So as you guys know, there are a lot of AI in healthcare courses right now, right? I mean, tons of it. It's about getting doctors to learn AI, but there's really not as much biology and health for computational scientists. I feel I've always felt that. I've always talked about it in other contexts. But I have Lewis here. Lewis come from probably the most AI informed cluster right now. Sorry, I know you just came back in. Just wanted to maybe get you to share about, this issue of getting both sides to care about, breaking down the silos and all of that. Yeah.
Oh gosh, this is
Yeah, biology.
oh gosh.
You sprang this on me. Okay. All right. Yeah, sure. Okay.
You probably
Yes.
gone for tons of courses
No.
at HG, that's why you're here. We went for some of the courses, we learned some stuff, but we don't like we say, we don't actually know what it means.
Yes.
So how do we kind of is it working how we are getting the clinicians to learn AI? Is the current model working? If not, how do we get it better? And then on the flip side, how do we get the scientists to learn the other side of it?
I guess it's always a matter of lingo, unfortunately, which you and I know. As much as we try to learn, we are never the guys. And we can only we are the false pretenders. That's why I always say that and I guess it's us all trying to role play each other and hopefully to come to a understanding. And then of course the real experts who are the computational biologists, the bioinformaticians, and perhaps you may also want to consider role playing yourself as a physician with a patient in front of you, or with a cohort of patients with a question. And I guess if we put ourselves in each other's shoes, and then and I think perhaps we will have a better appreciation of the scope of the challenges that we each face on our plate. And because it is never possible to have a clinician to replicate the role of computational biologists, and likewise vice versa for one of the guys who's actually doing the most intense and mind-boggling work to put themselves in the shoes of a physician with a patient. And I think this is how we have actually moved on. And how many guys actually have got triple degrees and double degrees? It's not possible. So, and that's where I think forums like this helps. And I face a lot of problems because for those of us who are actually doing genomics, the enormity of the data set is one that we always grapple with. And I do it after I put my children to bed at 10 o'clock. And that's not the best time, but I'm always asking if there is a better way to sort of alleviate this kind of backbreaking work to something that is more, I would say, more systematic. I do it when I'm not awake. So that's where I hope that helps for us when we are actually have got something more basic. I think for us, when we have a patient at hand with something that's more complex that some of you other guys actually may face. Yeah, thank you.
Okay, I guess we probably should try and call this to a bit of a close. Thank you, Rebecca. Any other feedback about this section on education? So it sounds like for this section, we all agree that maybe We need to look at both directions. But I just want to put it out there. Anyone has other sort of suggestions that we haven't necessarily, we talked about joint training, joint programs, probably getting people to get to different workplaces, internships and all that kind of stuff. But to me, that still feels like very much what we've been doing. If there's any other suggestions, you know, feel free to, maybe we can put up our slide. So we're meant to wrap up and give the way forward. So thank you very much again, everyone, for being here. And for staying very engaged and giving all your contributions. You know, it helped that it rained. You couldn't be doing something more fun elsewhere. But our contact details are up there. The reason why we put it up there is this is the end of the workshop, but it's definitely not the end of the feedback session. So I think we are supposed to write 3 to 5 pages summarizing the experience and then your insights and inputs into this thing for NRF. But please, 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 like if you go home today and you said, oh my gosh, there's this other thing I really want to send across, and I didn't get a chance to say it or tell us about it in your own groups and all that, please just email us using somewhat of a similar framework. Don't just tell us this is a problem you want fixed, because we're not going to have the answers. But if you can tell us this is the challenge, same thing, this is the challenge, this is what it can achieve, what AI models can be useful for it, why we can't do it now, are we the right people to do it? We will be very sure to try and incorporate it.
And we have data.
And which data sets will that come from? I mean, we're being very practical here because in 6 months, there's basically going to be a call for proposals, right? And so we really want this to be something useful to the community and all that. So please help us along on this way. Yeah. Millie, anything else?
Yeah, I just want to thank you. And if you have any questions, just send us emails, and anything related to this or something else you think that we can do together, I'll be more than happy.
Yeah.
Thank you all.
I want to thank the PhD students, the postdocs, and also Clarester here, who's had a very difficult job manning the Zoom and the slides. So can we give a hand to the rest of the folks? All right, I think that's it. Thank you, guys.
Thank you.